Hello students, here we will discuss case-based questions from chapter 2 to 6 which are very important for your first term exams. This is first case-based question. Read the passage and look at the diagram. It is the schematic representation of spermatogenesis. It occurs in seminiferous tubules of testes and begins at puberty. After reading this, now answer the given questions. Question number 1. Which cell division takes place during multiplication phase? See the diagram carefully. In multiplication phase, the primary germ cells, spermatogonia, divide several times mitotically and produce large number of spermatogonia. So, option A, mitosis is the correct answer, which takes place during multiplication phase. Question 2. How many chromosomes are present in secondary spermatocyte and spermatids respectively? In the figure, K are secondary spermatocytes formed by first meiotic division of primary spermatocytes J in the picture. As a result, number of chromosomes reduced to half. Further, secondary spermatocytes undergo second meiotic division and form spermatids, which are L in the figure. So, both secondary spermatocytes K and spermatids L have half the number of chromosomes. Option C is correct. Both have 23 chromosomes. So, mark C as a correct answer. Question 3. Transformation of L into M is known as transformation of spermatids L into sperms M is called as spermiogenesis. So, mark B as a correct answer. Question 4. Select the correct option. Out of 4 given options, option C is correct. Spermiation is the release of sperms from seminiferous tubules. This is the correct statement. After spermiogenesis, sperms had become embedded in the Sertoli cells and finally released from seminiferous tubules by the process called spermiation. So, mark C as a correct answer. Question 5. Which hormone acts on spermatogonia to stimulate sperm production? Pituitary hormone FSH or follicle stimulating hormone acts on Sertoli cells and stimulates secretion of some factors which help in the production of sperms. So, here mark D as a correct answer. Case-based question number 2. This is related with development of embryo in the embryo sac of monocotyledons and dicotyledons. Read the complete paragraph, analyze all the information and then answer the given questions. Question number 1. The true embryo develops as a result of fusion of. In double fertilization, out of 2, one male gamete fuses with an egg cell to form diploid zygote and this zygote develops into the embryo. So, correct answer is option B. True embryo develops as a result of fusion of an egg cell and a male gamete. So, mark B as the correct answer. Question 2. Which of the given statements are true? Statement number 1. During the development of a dicot embryo, heart-shaped embryo is followed by globular embryo. This is wrong statement. Actually, during development of dicot embryo, first globular embryo is formed and then heart-shaped embryo is formed. Statement 2. The part of embryonal axis above the level of cotyledons is epicotyle, while the part below the level of cotyledons is hypocotyle. This is the true statement. Epicotyle terminates with plumule or stem tip and present above the level of cotyledons, while hypocotyle terminates in radical or root tip and present below the level of cotyledons. Now see the statement number 3. Monocot seeds possess a single cotyledon represented by scutellum. This is also correct statement. So option B is right. Statement 2 and 3 are the correct statements. So mark B as a correct answer. Question 3. Refer to given diagram of the embryo of an angiospermous plant with parts labeled P, Q and R. Select the correct statement regarding this. See the diagram and read the statement number 1. Part B supplies nutrition to the developing embryo. This is correct. P is scutellum, which is a cotyledon and supplies nutrition to the developing embryo. Statement 2. Part Q is the protective sheath of radicle and root cap. This is wrong. Q is coleoptile जो शूट एपेक्स एंड लीफ प्राइमोर्डिया को प्रोटेक्ट करता है. स्टेटमेंट 3. पार्ट आर इज द प्रोटेक्टिव शेथ ऑफ शूट एपेक्स एंड लीफ प्राइमोर्डिया. दिस इज ऑल्सो रॉन्ग. आर इज कोलियोराइजा जो रेडिकल एंड रूट कैप को प्रोटेक्ट करता है. स्टेटमेंट 4. द एम्ब्रियो शोन इन द डायग्राम इज present in members of family Poaceae. This is also correct. So, two statements are correct. Statement number 1 and statement number 4. So, mark C as a correct answer. Question number 4. Consider the following parts of an embryonal axis of a dicot seed: hypocotyle, epicotyle, radicle and plumule. In which of the following the above parts are correctly arranged from top to base? In option C, the parts given are correctly arranged from top to base. Number 4, plumule is at the top. Number 2, epicotyle is next to plumule. Then number 1, hypocotyle, and at the base is the number 3, radicle. So, mark C as a correct answer. Question number 5. In grass family, the cotyledon is called option C, scutellum. In grass family, cotyledon is called as scutellum. So, mark C as a correct answer. Case-based question number 3. The given information is about translation process where chain of amino acid is formed from messenger RNA, and in order to code all the 20 amino acids, codes are present in the form of 3 nucleotides. After reading the information given, answer the questions. Question number 1. What is codon? Option D is correct. Codon is a part of messenger RNA molecule that has a sequence of bases coding for an amino acid. So, mark D as a correct answer. Question number 2. 3 consecutive bases in the DNA molecule provide the code for each amino acid in a protein molecule. What is the maximum number of different triplets that could occur? The triplet code consists of 3 out of 4 nucleotide bases: adenine, cytosine, guanine or thymine. Thus, the maximum number of codons is 4 to the power 3, which is equal to 64. So, mark D as a correct answer. Question 3. Listed below are some amino acids and their corresponding messenger RNA triplets. Check the table for this. Now, you have to tell which DNA sequence would be needed to produce the following polypeptide sequence. और polypeptide sequence है alanine, arginine, lysine and phenylalanine. See the hint for getting the answer. पहले messenger RNA पर इन चारों amino acid का codon sequence बनाइए. It will be alanine GCA, arginine CGA, lysine AAG and phenylalanine UUU. Now, Find the sequence on DNA. The complementary bases of this messenger RNA will be CGT, GCT, TTC, AAA on the DNA strand. This sequence is given in option A. So, mark A as a correct answer. Question number 4. Identify the nonsense codons among the following. Nonsense codons are UAA, UAG and UGA, which do not code for any amino acid and are also called as stop codons. So here option C, UAA is a nonsense codon. So mark C as a correct answer. Question 5. A polypeptide is made using synthetic messenger RNA molecule as shown. टेबल देखिए, सिंथेटिक मैसेंजर आरएनए जो यूज़ किया गया है इसका सीक्वेंस है UUU, AAA, UUU, AAA एंड जो पॉलीपेप्टाइड बना है उसमें एमिनो एसिड्स का सीक्वेंस है फिनाइल एलेनिन, लाइसिन, फिनाइल एलेनिन, लाइसिन. आपको बताना है व्हाट आर द DNA कोड्स फॉर द एमिनो एसिड्स. Option A is correct. The triplet codon of phenylalanine UUU will base pair with AAA in the DNA and that of lysine AAA will base pair with TTT. So mark A as a correct answer. Case based question number 4. This information is about assisted reproductive technology involving transfer of gametes into oviduct. This technique is advised by the doctor to a young couple who is unable to bear the child. Now, answer the given questions. Question number 1. Identify the technique adopted by the couple. Option C, GIFT technology or gamete intrafallopian transfer technology is adopted by the couple, where transfer of ovum collected from the donor into fallopian tube along with man's sperm is done. So here mark C as a correct answer. Question 2. What is prerequisite to this technique? The prerequisite of this technique is fallopian tubes should be normal, because gametes are transferred into fallopian tubes. So here correct option is B, mark B. Question number 3. This is assertion reason type question. See the assertion statement. GIFT is an in vitro fertilization technique. This is wrong. In GIFT technique, fertilization is in vivo. It is not in vitro. See the reason statement. In GIFT, gametes are fertilized inside female body. This is the correct statement. So here mark D as a correct answer. A is false but R is true. Question number 4. The procedure advised to couple involves the transfer of, option C is correct. In GIFT technique, both the sperm and unfertilized ovum are transferred into the fallopian tubes. So mark C as a correct answer. Question 5. What is the success rate of this procedure? Option D. The success rate of this procedure is 27 to 30%. So mark D as a correct answer. This is case based question number 5. This case is about DNA replication in eukaryotes and in prokaryotes, bacteria and viruses. Read it carefully and answer the given questions. Question number 1. In viral DNA, how many origin of replication are present? Replication begins at a particular spot called origin of replication or ori. Bacterial and viral DNA has a single origin of replication. So mark A as a correct answer. Question 2. Select the main enzyme involved in replication. A large number of enzymes are required for DNA replication. DNA-dependent DNA polymerase is the main enzyme which takes part in combining deoxyribose nucleotides to form new DNA strand. So here mark C as a correct answer. Question number 3. Read the given statement and fill the correct option. Enzyme dash act on the ORI site and unwinds the two strands of DNA by destroying dash bonds. Option B. Enzyme helicase acts on the ORI site and unwinds the two strands of DNA by destroying hydrogen bonds. So mark B as a correct answer. Question 4. DNA strand built up of Okazaki fragments is called. Option A. Lagging strand. Lagging strand is a replicated strand of DNA which is formed in short fragments called Okazaki fragments, and its growth is discontinuous. So mark A as a correct answer. Question number 5. Select the incorrect statement about DNA polymerase in eukaryotes. Actually, there are 5 types of DNA polymerase: alpha, beta, gamma, delta and epsilon. Now, see the functions of these given in options below. Option A. Polymerase alpha is required for initiation of replication. It is true. See the B. When the RNA primer gets removed, the gap is filled by DNA polymerase beta in eukaryotes. This is also correct. Beta DNA polymerase RNA primer को remove करने में help करता है. Now see the C point. Polymerase epsilon helps in elongation of lagging strands. This is also correct. Polymerase epsilon lagging strand के formation and elongation में help करता है. Now come to point number D. Polymerase delta is largest and main enzyme for DNA replication in eukaryotes. This is wrong. Polymerase delta leading strand के elongation में help करता है and polymerase alpha is the largest and main enzyme for DNA replication in eukaryotes. So here incorrect statement about DNA polymerase is D. Mark D. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.